the technical difficulties before the powers that be stole our Zoom meeting. And just talking about the outline of the presentation, then I'm going to talk about some issues with the measurement of root hydraulic conductivity or conductance to start. And then I'll move forward and talk more about biological things. But I think it's important to talk about the measurement because of there's some biology in there. We have some artifacts, and I want to introduce people to a couple of them. But I think there's also some biology hidden in there. So when we got cut off, I was on this slide just kind of introducing the measurements, right? And so we're talking about flows, how flow changes as a function of a pressure changes. And this is what a conductance is. So in a root system, we can normalize this to get a conductivity. Typically, it's done with a root down here in the corner of this image with a surface area, right, to get this thing called LP. People normalize root hydraulic conductance by all sorts of stuff. And in the literature, that can be a little confusing. So it can be confusing in general because you have conductance that gets normalized by a bunch of different things. It could be normalized by like shoot biomass or by root biomass. But also you can use different gradients to drive the measurement. So you can use an osmotic gradient versus a hydrostatic gradient. And again, this morning, I don't want to take a lot of time getting into the details of these two things, but this changes our interpretation of what we're actually measuring. And then we can have the measurement be on different things, the whole root system. It could be on even root portions or individual roots. But the caveat here is that no matter what you're going to measure the conductivity of, the flow rate has to be high enough to be measured, and you have to have an instrument that's sensitive enough to measure really low flow rates if you're going to do it on, say, an individual root. And this can become limiting. Okay. So the root hydraulic conductivity and the problems or the issues that I want to introduce this morning, I think stems from the fact that pushing water through like complex cellular tissues is a lot different than pushing water through a xylem network that has a lot of big open pipes, essentially. So with stems, say we're doing a stem measurement, you get a lot less artifacts on a stem. And the measurement is much more stable, and everything tends to work better. But when we're going across a tissue, and I'm going to show you an example from a berry pedicel too, or we have a tissue that's pretty complex. It doesn't have a lot of xylem, but it has a lot of other tissue area. I think we start pushing water through this space, things get kind of weird. And we're not really sure anymore where the water's actually flowing. This is from this book chapter that we wrote recently. And you can see we made this figure. This is the classic composite transport model figure. And you can see here that you can have an apoplastic pathway. You can have this is the apoplastic pathway. I think you can see my mouse now. Or a cell-to-cell pathway. Things could go, this is the Casparian band. They could go transcellular or maybe through the band. I'll talk about that briefly, or through aquaporins. But the bottom line is all these pathways, they coexist. It's not as if the water's just flowing through one. It's just flowing through all of them as a function of the resistances. So this gets kind of complicated. So traditionally, when you make these measurements, before you get a number that's like my conductance is whatever it is, you build up a pressure flow relationship. And that's what I want to start with, is just talk about this relationship briefly. That there's this assumption when we make these measurements that this relationship is linear. So I just took this example just randomly from a plant physiology paper that was in Plant Phys, and just to show you an example. And in this example, the authors took, they're using a hydrostatic suction pressure. They measure the conductance. Each one of these things is a root or root system. They get a flow, and then they increase the pressure. They get another flow. And then they basically regress these 2 points, in this case 2, pressures, which I'll say at the outset, I don't think is enough, right? 2 points makes a line. But then in this case, they also force it through 0. This is an interesting choice. I don't know why they would force it through 0, because in root systems, the balancing pressure doesn't have to be 0, right? Root systems or roots, they often have a root pressure. And I did something kind of poetic license. I just took their graph and drew lines through the points. And that's what you see on the right. These are if the lines were just drawn through the points and not forced to 0. And you see that this changes the result. Maybe not significantly, but I'm just using this as a point here that, all of a sudden these points don't go through 0 if you just regress them through the 2 points that they measured. The other thing is sometimes you would see that, these 2 lines down here, they become pretty much parallel, right? And of course, this relationship between the pressure, the applied pressure, and the flow, the slope of this line, that is the conductance, okay? And then if we normalize that, that's a conductivity. So that's the first thing. This is the measurement. You build up these relationships, and then we get, from the slope, we get the conductance. So this, and we always assume this thing is linear. This is a paper that I was the first author on from 2012. And on the left, this is in Grapevine roots. And I'm applying a pressure here, right? And then I'm measuring the flow rate. And in this case, we measured it at 5 pressures. Okay, so you apply a pressure, flow, and then you regress this, we get a conductance. It can change or whatever. But I want to point out that and just like everybody else, just made these things linear, right? And let's contrast this with the study of the graph on the right. So this is a study that I was also a co-author on, but was led by this guy, Torsten Knipfer, who's done a ton of work in roots and in other systems in hydraulics. Torsten is just a master in this stuff. So Torsten was working on grape berry pedicels, which are pretty complicated structures. And he was looking at hydraulics in pedicels. And what you see there is that it's got like this 2-phase sort of pressure flow relationship, where when Torsten put in low pressures or even negative pressures, he was using a root pressure, like he modified a root pressure probe, for those of you who know what one is, to do these measurements. And he gets basically 2 phases. One phase down here in the lower region, and then all of a sudden it gets steeper in these higher pressures. So this kind of creates a conundrum directly. It's like, okay, if I take this slope, I get 1 conductance. If I take this slope, I get another conductance. So what do? In this work, we reported both. And we called them different things, okay? But nevertheless, this does create issues. And what I did was I just box here for clarity, the pressure ranges, and I superimposed those back onto this earlier study that I did in roots. And you can see I didn't even measure pressures in that range, in that study. So I'm assuming these linear things. What I measured maybe was the equivalent of this conductance. When you go back, and I've done this was years ago, when you go back and you do more measurements on roots, what we found is that this case on the right is oftentimes how it is. That you see it's either got 2 phases or it's non-linear, okay? In that at very low pressures, you get 1 conductance and then it ramps up. So this assumption that the pressure flow relationship is linear is only true across certain pressure ranges, okay? So we have to be careful about that. And then that immediately, though, raises an interesting question, which is, how come? Why is that? And this is a cross-section, again, from Torsten's paper on berry pedicels. So he's pushing water through a berry pedicel. And I'm just showing you autofluorescent cross-section of a pedicel here that was published in the paper. It's really a pretty cross-section. And you can see this is the xylem area in the blue. These are the phloem caps out here. And what you can see is that, okay, when you push at a low pressure, we're going to assume that you go through the xylem, okay, predominantly. But when you get this shift, when the conductivity all of a sudden goes shing and it gets a little bit steeper, where is that coming from? What's going on there? So one thing that wasn't published in the paper but is discussed in the discussion, Torsten and I were working pretty closely with each other on this project, is that we would do from time to time dye infiltration experiments along with the measurements. And when you get at higher pressures, sometimes normally what you see is that the xylem are all stained with dye, meaning you're pushing flows through xylem. This is what you would expect. But when you get to the higher pressures, sometimes you get weird flow pathways. You get them going through tissues. If you just interpret this as the dye going through spaces, maybe it's apoplastic spaces between cells, we would see maybe staining around the caps out here. And so this makes you think, okay, maybe this higher flow is an artifact, okay? But maybe it's not. It depends on what the physiological relevant pressure gradients are. But I'm just bringing this up as one of the difficulties of this measurement. It's something to think about, especially in complex tissues. I will say one other thing. I'm going to go back. In that in root systems, not only do you get kind of a biphasic sort of ramping up, you also, if you apply enough pressure, and this is experiments that we did at Davis, again, unpublished, it also saturates at some point. It rounds off at the top. So if you were to do a full range of pressures from extremely low to extremely high, I think what you see is a sigmoidal sort of relationship in the pressure flow, which is a little bit different than we would expect. So is it linear? Sure, but only across certain pressure ranges. So the second really major problem is this thing that I'm just referring to today as wounding decay. And this is the problem that the moment that you cut a root off or a root system off from the plant, you've intrinsically changed it. This has been a huge problem in hydraulics in general. This caused all sorts of pain for people doing hydraulics, studying embolism, studying all sorts of things. And it also is a pain for people studying roots. This is a great treatment of the subject. It's in this paper by Steve Tyerman's group, where they just look at the wounding decay of root hydraulic conductance. This is in soybeans in this study. And you can see in this graph on the left how fast it happens. So they basically detopped the plants and then measured root hydraulic conductance. And you can see that within minutes, the half-time here is 5 minutes, you get, say, an 86 or 71% decline in the value. So this means that it's very difficult to capture the real values. On the right, they did something that was cool. They also said, okay, this is the first hour, right? And we get these declines on the left, the first hour. But what happens if we wait 1 hour, 3 hours, 5 hours, 7 hours, 24 hours? This is the graph on the right. And what they saw was in soybeans that this stays very stable. Now, I've also done these experiments in our lab. I had a PhD student do these experiments, and I can say that in grape, this is not what we found. In grape, it declines forever for as long as we measured it. So in grape, at 1 hour, it goes down. At 3 hours, it goes down more. At 5 hours, it goes even more. At 7, more. If you wait overnight, come back at measure it, it's almost near zero. So in our hands and in our lab and with grape, you don't get a stable phase after the initial declines. Somebody needs to mute their mic. So I just wanted to bring up these two issues as a way to kind of talk a little bit about best practices. If you're out there, maybe it would be great if you muted your mic. It would just be helpful for me so I don't get feedback in my ears. So until we have better ways of measuring the fluxes and the pressure gradients in a non-invasive way, we need to be really careful about making these measurements. We need to make the measurements with an absolute minimum amount of time between when you perturb the plant and when you make the measurement. That's hard. That's really technically difficult because these measurements require oftentimes connecting things, making sure there's no leaks. That is a big challenge. But also we need to remember that the pressure flow relationships, especially through roots, they may or may not be linear. So we need to make measurements across multiple pressures. I mean, I think that this means like more than 5, maybe 5 minimum to draw, you're regressing a line through them. This is laborious, right? And it takes time. So then we kind of get back to the first problem, right? We can't take too much time because we have this decay. And then also probably the lower the pressure, the better, okay? But this last point that I make on this slide, I think is the most important, and it's the one that I'd like people to maybe remember, is that what you really need to do is not like read a paper and go, oh yeah, that's the way these authors measured it, that's the way I'm gonna do it. What you really need to do is do a lot of upfront work with whatever system you're gonna work with, do a lot of different kind of like, you know, exploration sorts of studies to see, you know, assess how much wounding decay am I getting, you know, is the pressure flow relationship linear across the pressures that I'm going to use, and then kind of have an idea of how your system or your plant is behaving before you get into making a lot of measurements. Okay, so this starts off, I'm going to move quickly from my hate to the love portion of the presentation. So be really, really careful with the measurements. The measurements are really problematic. And the bottom line is we're probably always grossly underestimating what the conductance is because of how rapid the decay is when we cut it. So let's talk now, though, about biology. So in a grapevine, it's a perennial root, right? So its development is really quite different. And the first subject I want to talk about is this idea that the root becomes impenetrable, or that suberin plays a huge role in limiting water uptake in roots. And this is from a paper we published in 2013 when I was working with Andrew Macaron and with Torsten at Davis. And this is just a schematic of a grape root, and it's kind of interesting. So I always think of it as having basically 2 phases, right? It has this phase when it has an exodermis and a cortex. This is kind of like the young root. And what some people may not be aware of is the cortex and in perennial roots, the whole cortex and exodermis, all these outer layers of the root, at some point they slough off. They deteriorate and they fall off of the root. Because within the root, on the inside, the periderm basically starts to arise. This is what's eventually going to become the woody portion of the root. And in this work, we basically go through and look at the different anatomical transitions in the root. This is a schematic of that. And with that comes major changes in the suberin structures in the root. So in this lower panel, what we have is berberin. This is a fluorescent dye for suberin waxes. It's called berberin hemisulfate. It's a very classical sort of visualization. And you can see these are the different portions of the grapevine root. You have the tip, okay, and we're just going through development. This is where the xylem starts to develop. This is the exodermis here. This is a zoom into the exodermis, and we just go like this, and eventually we get to the point where then the periderm takes over. The cortex in this picture has sloughed off, and this zoom here, the periderm is like the sandwich structure of suberized cell walls, basically. And what we found in this, the work was basically a very kind of went hand in hand with the traditional common wisdom, which is that root hydraulic conductances in the tip portions that are unsuberized, they are higher. And this correlated with all sorts of stuff. It correlated with higher levels of aquaporin activity and expression. And, you know, there's less suberized structures. And then you get a decrease in hydraulic conductance in the older root portions. So to some extent, you know, okay, there is a decrease, but we never found along in fine roots sealing, meaning that even in the older root portions, they were still conductive. And so I think that maybe when it comes to roots, there's a lot of textbook received knowledge that there may be some common misconceptions that we have. One of these misconceptions may be that this idea that suberization really can decrease hydraulic conductivity of roots, and that the Casparian band forces water across membranes. It may, to some extent, increase the resistance, or in other words, decrease the conductance, but does it seal it? I personally don't think so. So I like this work, and I wanted to share it with you guys today. This is kind of an old work by Stoidla's lab from 1993. I don't know how many like pure like hydraulics people are out there. I'm not even a pure hydraulics person, by the way, out there in the audience, but Stoidla is very well known for root hydraulics, incredibly prolific in the subject. And his papers are really hard to approach. Like they're full of like these pressure relaxation curves and super ultra technical articles. So I reworked the data for our discussion today because it's so much easier to see. He did this experiment that we may consider to be kind of crude, where they were interested in exploring the role of these suberized structures. And this is in corn. And they did it kind of in a crude way where they physically pierced the root, or they physically even, this is an example I have in the slide here. I put in here an example of the minor wounding treatment. That's these bars on the left of the graph. There was a major wounding treatment though. And in that treatment, they like got in there with a razor blade and like scrape off root structures. And this is pretty brutal, right? But the result is kind of interesting. What did they find? They did these manipulations to ask the question, okay, if I kind of wound the root, if I pierce the endodermis, if I kind of basically create a hole through these suberized structures, what do I change? Do I change hydraulic conductivity really drastically? And what we see is they were focused on two things. One was the amount of root pressure that the root was producing, and the other was hydraulic conductivity. In their work, they're expressing this as a half time. So let's just wrap our heads around this. If you reduce the half time, it means the conductivity goes up, okay? So what they found was when they minor wounded it or major wounded it, on this axis, it's the percent reduction in whatever it is, either root pressure or the half time. They really, really affected the root pressure. So this pointed them to think that, okay, when I really kind of mash the root or really mess with these structures, I'm really changing ion flow in and out of the root. Almost 100% reduction of root pressure in the major wounding. But when you minor wound it, they didn't see much of a change in root hydraulic conductance. It was like about 10% reduction. And it goes up in the major wounding. So it looks like there's some contribution. This is fuzzy, right? It's fuzzy, but it definitely, one thing it says to me is that maybe when we think about these structures, their effect on ions may be much more important than their effect on water. And this is interesting to me. So they wrote in their paper this, which is, these results indicate that the endodermis is not a particular barrier to the movement of water. This is a quote from their discussion in this paper. And we conclude that the barrier is spread evenly over the living cells of the root. I think that that's a great line. I like that line a lot. That it's kind of silly of us to focus on these like single cell layer structures. They may be important, maybe for other reasons, maybe an ion uptake or controlling root pressure or something. But when we think about how roots take up water, I think that this is a nice way to look at it. This idea that it's spread evenly across the tissues. Then there's this other work by Schreiber, and Steudler is a co-author on it too. This is 10 years later. Really like intricate work on suberin. And it's 10 years later, right? I like this in science. 10 years go by and quote, the deposition of suberin in root cell walls do not allow straightforward conclusions to be drawn regarding the degree of inhibition to water and ion transport. So I would argue that we're still there. You know, this was in 2005, it's 2020. We're still trying to figure this out. And the thing that kind of is humbling to me is that we're really limited by the biophysics. We don't understand the biophysics. I can do a lot. This is a manuscript that we just submitted for publication like a month ago from our lab, looking at suberin and grapevine roots. And I feel like we did a lot in this study. We did, I'm just gonna walk you through really quick in one slide what we did, okay? We visualized changes under water deficit. This is kind of a very qualitative thing, again, with berberine hemisulfate staining. You can see suberin lamella and other structures get upregulated under water deficit. This has been known. We characterized all of the chemistry at a really fine level in different root portions. This is the root tip, primary xylem. This is secondary growth, early and late. So we looked across the length of the root, and we see that under different water stress levels, either medium or high, you get this global upregulation of suberin everywhere in all these structures. We did a big RNA-seq study, and we identified these orthologous transcription factors that in Arabidopsis, these things control, in part, there's multiple transcription factors that do it, but they're one of the cornerstone regulators for suberin biosynthesis. And then we isolated the orthologs of those in grape, and then we showed that they indeed upregulate suberin biosynthesis through transient expression in tobacco. These are all really classical sorts of approaches. And then in the end, though, we're still only correlating. So I like to do this sometimes in talks, is like, it's kind of like throwing down the gauntlet. If there's other people who are interested in doing root hydraulic research out there, that there's so much more to be done. We're still really only correlating. I can characterize this, but I still don't know what the impact is, and still until the time when we're able to actually manipulate it in situ. And so hopefully that's the direction we're going. We're going in the direction where we can leverage some of these transcription factors to then start to turn suberin up and down, and then measure how it affects physiology. So that's what we want to do eventually. But biophysics really is the limiting factor. I was trained, when I, at Davis, I was actually trained, I got my undergraduate degree in molecular biology. So I started off as a molecular biologist, and I kind of like then later changed over to physiology. The more I got into it, the more I was like, oh, you can do a ton with molecular biology, but then at the end, I don't understand what's going on still. You know, this is kind of a bummer. So we got to get at the physiology. So changing gears a little bit, and talking about root hydraulic conductivity, one of the things to always remember about it is it's just constantly changing. It's like really analogous to something like stomatal conductance, where it's hard to capture it. When a physiological phenomena is changing really fast in time, our measurements tend to be bad, I think, because we're constantly trying to stop it. You know, stop, so I can get a measurement out of you. Root hydraulic conductivity is indeed has to be like this too. This is, again, going back to Steve Tyerman's group in Adelaide. It's a nice example from grape, where you can see that this is on this axis root hydraulic conductivity. You can see that it's diurnally regulated. You know, it goes up, you know, to midday and then goes back down. And then if you look at the relationships of the conductivity with some other, you know, flux related whole plant thing, in this case transpiration, you can see that it's noisy. It is indeed noisy, but it's pretty positive and it's strong. So this is just something I like to just bring to everybody's attention, that there is this really close integration between the shoot and the root. And this integration is occurring in real time every day. And this makes measurements, I think, of root hydraulic conductivity very problematic and very noisy. It's also kind of just cool because the plant is basically, you know, every day upregulating its root hydraulic conductance to compensate for these increased fluxes. So in essence, and I'll get to this a little bit later, what is it doing? It's basically controlling the pressure drop across the root system. So if I'm increasing my root hydraulic conductance, I can decrease that pressure drop, and I can basically make these bigger flows result in lower tensions in the plant. So this is just one thing I wanted to bring to everybody's attention. So turning to water deficits. So traditionally, you know, water deficits, they always result in the long term in a reduction in root hydraulic conductance. But the timing here, going back to this idea that if we go back and we just like put this thought in our head that, okay, this is a really dynamic thing that's coordinating with above ground gas exchange, the timing of what goes on during water deficit and root hydraulic conductivity is pretty vague. I think that's mainly because, going to the third point here, because most studies are categorical. This is a classic study by North in this little agave sort of little succulent here. And it's measured like this, right? We have a category where we have well-watered plants at minus 0.1 megapascals, soil water potential. They get stressed. This was in this particular study, it was a 10-day stress for the amount of time. That's a long time, 10 days. And then, you know, a rewater treatment. I'll introduce just this really briefly, that a lot of the times in these studies, you'll see inhibitions, quote unquote, with things that are going to inhibit aquaporins, because aquaporins, of course, contribute to root hydraulic conductance. So in this case, they use mercuric chloride. That's what the hatched bars are. I put a little down regulation here. This is due to aquaporins. That's what we're going to contribute this to. These inhibitors are pretty nonspecific though. So they're changing kind of global water relations and transport across membranes. So they're also pretty crude. We're still, the science is very young here. We're in a very crude approaches, I think. So if we interpret just this top panel, let's just walk through it for a second. If we interpret this traditionally, we would go, okay, under well-watered conditions, this is what the root hydraulic conductivity is. If I inhibit that with mercuric chloride, it goes down, boom. Okay, and that reduction, whatever its size, we're going to attribute that to, let's say, membrane processes. I want to be vague about this, but traditionally someone in their paper would say that's what's due to aquaporins. Now, I water stress the thing, and okay, I get a reduction in this. There's no more, I can't inhibit anything else. When I try to do an inhibition, there seems to be no contribution here of any aquaporins or anything. And then I rewater it, and it comes back. And when it comes back, then I can inhibit it a little bit again. So if I just, all things created equal, I would say, okay, water deficit downregulates this in a reversible manner. And it's mainly aquaporin driven, and it can come back. This is different root portions, okay? But there's a lot of different mechanisms that happen simultaneously or at different stages, and this is where I want to kind of focus on trying to get more out of the timing. when we look at the reductions in root hydraulic conductance underwater deficit, you have aquaporins that get regulated. You have changes in these suberized structures, which is what I was just talking about in the preceding slides. And then you also get other structural changes, which I'm going to talk about next. You get this lacuna, and this is work that Idolo Cuneo and Andrew McElroom's lab has done recently that's been pretty awesome. And this may contribute to root shrinkage or just the roots basically deconnecting from the soil altogether. But in general, we need to get away from having categorical treatments and move towards kind of other ways of looking at data. This is one of these Cuneo studies, kind of like this, where instead of categories, we can look across a whole range of water potentials. And then we can start to understand a dynamic. In this case, the work was focused on these gaps in the cortex. So on the right, I'll draw your attention to the micro CT images on the right. And what they did was that these are images that are taken in roots using micro CT. The roots are intact, right? So this is using X-rays to scan in 3 dimensions roots. And then they can understand what's going on anatomically within the root in real time without cutting the root up. And this has been a really powerful technology that a lot of people have utilized for studying embolism and cavitation, and more all sorts of other processes in plants. And you can see on the fine root panel that as things become more stressed, the cortex, which by the way, eventually during development sloughs off and is lost anyway, starts to gather these huge gaps. And these were traditionally called lacuna. And so you can see them form. And so what they did, what Andrew's lab did, is they basically quantified the dynamic of that in relationship to the drops in root hydraulic conductivity. So on this axis, we have the increase in the percent of those gaps. Those are the triangles. The black dots are the decrease in the root hydraulic conductivity. And this allows us to get a much better feel than a categorical study that, say, if we just took stressed and unstressed. Here we actually see timing. And we see that things are happening. There's this lone little point down here at 0. So it's coming up like this. Now, one caveat I have, one kind of like, this is just a really small thing, but I'm gonna kind of push this graph a little bit. The scale on the right is from 0 to 35%. That's a little odd, right? It's the percent of the cortical area. It should probably be on a 100% scale. So I'm just gonna go like this and put the points in on a 100% scale. So if you took these triangles, you can look at them either way. I'm not sure what the correct way to interpret it is, but it changes how we interpret the timing a little bit if we put it on a 100% scale. But one thing you can see for sure is that the drops in root hydraulic conductivity are really abrupt. It's almost going straight down. And this suggests that it's happening really, really fast. Okay, so this is a really nice study. If anyone's interested, I encourage this one and a recent one that I'll cite at the end. We have done similar measurements in our own lab. This is from a PhD student, a former PhD student of mine, Li Zhang, and this is 2 different rootstocks, and I just am showing you all the data here looking across water potentials. And you can see also that it really looks like changes under water deficit happen really fast. So things go down really, really abruptly. And I want to remind everybody at this problem that we have with the measurement at the outset, which is this idea that when we wound it, we have these instantaneous drops in the conductivity anyway that we're probably missing. This means that we probably are underestimating these values. And so these could be even higher. The drop could be even quicker than we think. And there's no reason to think that wounding decay should act the same depending on all water statuses. It could be that under well-watered conditions, you get way more wounding decay than you would under water deficit. This could be because aquaporins have a disproportionately large contribution under well-watered circumstances. There's reasons why you would think this. But anyway, regardless of these caveats and these difficulties with interpreting what we're looking at, it looks like the changes are really fast. So this is just part of my goal with the seminar is to also stimulate people to think about different things. So sometimes I just threw something in here. You know, we tend to focus on root hydraulics as controlling fluxes, but also don't forget that you can also think of it in terms of controlling pressure drops across the root and in the plant. So I really encourage people, if you're interested in the subject, to check out this paper. I like this paper a lot. It's The Significance of Roots as Hydraulic Rheostats. It's in JXB, published 10 years ago by Christophe Morel and company. It's a really nice kind of primer, kind of get people thinking about looking at things in a new way. But until we have real-time data in intact plants, the whole story that I'm discussing will remain largely a mystery. And going back to the love-hate theme of the seminar, this is like kind of where I'm at personally, is I really would love the field to come to this. And because I know that until we come to this, we're going to have a lot of difficulties making progress. How do we do that? It's a huge challenge. So I'm going to leave the seminar at the end here with just one last thought, just for interest. Maybe somebody will pick up on this and run with it. That this idea that hydraulic conductance changes along the length of a root and that root tips are the most conductive is really interesting. Buried in this paper that we published back in 2013, it's buried because it's like in supplementary data and we don't really talk about it that much in the paper, is we did this little modeling experiment in the paper where we said, okay, you know, you have different conductances, really high conductances in these unsuberized portions of the root tip that decreases as you go back. And we could imagine different scenarios, and we did in this thought experiment where either the rest of the fine root remains conductive or it doesn't. This is if it seals, it's a nonlinear scaling. And this means the root at a certain length, it saturates essentially, or it doesn't, it just keeps being more and more conductive. But regardless of what you do in these models, when you look at how water gets taken up in an individual root, what you would find is that it's really dependent on the size of these highly conductive root tips. And this pointed us in a direction of, okay, this is interesting. This means that there may be some intrinsic link just with growth itself, meaning that when roots are growing faster, they are more conductive because there's a larger proportion of the root is dedicated to these kind of highly conductive unsuberized portions. And when growth slows and or stops, that portion is very small relative. Is there, anything to this? Well, I don't know. I would like other people to do research on this. And so that's why I put it in my talk. We've done one experiment in our lab on it, and it's intriguing. So I'll share it with you. This is unpublished work from former PhD student Li Zhang. And what we did is we had these cylindrical rhizotrons and grapevines growing in them. And these things, by the way they're set up is they're these cylindrical rhizotrons that are like in a sandwich. So we could basically map the growth of roots growing along the surface, and then we could open the thing up like with a hinge, and we could harvest these roots out and pull them out. So we could pull them out and do measurements on these roots when we knew how fast the root was growing. There has to be better ways of doing this. So what we found was really intriguing, that when we measured LPR and we correlate that with the growth rate, it's really noisy. These are averages. These are the standard deviations ranges, which are huge. So I don't really know why it's so variable. Root hydraulic conductance is a variable measure already, and we're trying to kind of do something, I think, here that's pretty ambitious. But it's intriguing that we got these correlations to be so positive, and they're statistically significant for whatever that's worth. So this is something to think about. So in conclusion, I think the take-home messages for me are that, root hydraulics, they're really changing. They're changing all the time. They're really fast. And this poses a huge challenge for us when we're measuring stuff. It's probably one of the reasons why the measurements are so noisy. It reminds me a lot of gene expression analyses. Gene expression is a noisy measure too, right? Because these things are fluctuating in real time so fast, and they have a wide range. We really need to learn more about the impact of structural changes. And we're still mainly just correlating, is what I would say. But there's so many interesting questions that I'm just throwing this stuff out for fodder. Like ABA, by the way, over the very, very short term has been shown actually to increase root hydraulic conductance. And even there's some studies, even under short-term or mild water stress conditions, they see an increase, not a decrease in root hydraulic conductance. So this is kind of intriguing what's going on there. In our lab, we've done ABA treatments on roots, and we get the exact same results in grape. In our hands, under the short-term, ABA also increases the root hydraulic conductance of grapes. So is this like, there's some link here with turgor regulation of tissues or something? I don't know what's going on, but this is really interesting. At what point do reversible modes of regulation like aquaporins stop, and more drastic modes like lacuna or root shrinkage or even senescence, right? Because a lot of roots die seasonally in perennials. When do they take over? We don't know this really. And like, is reversibility something that even occurs? This is kind of like the idea, maybe if we want to use an analogy in leaves, if a plant's going to shed a leaf, the moment it decides to shed that leaf, it's not going to decide to bring it back, right? It's committed. So is reversibility something that it even employs in roots? Because remember that what happens in an individual root, doesn't make what happens in the root system. The root system is a population of individual roots, so it could be different. And I point you to this latest paper, again, by Andrew McElroom's lab in Cuneo, that they just published, which kind of speaks to this. And I just like to thank everybody who was involved in some of the research that I presented. Let's see, this is my former PhD student, Lee, and then this is Fred Domergue as a suberin chemist and specialist who I collaborate a lot with on the suberin work, and Isabel Merlin is a tech who does a lot of that work in our lab. This is Andrew McElroom and Torsten, who were basically with me the whole way at UC Davis for all the previous work. So that's it. This is my email address, and you can always feel free to email me. I love email exchanges. So thanks for listening, guys.
Thanks, Greg. It was great.
So does anyone have questions?
I have a question. I have some comments. Yeah? You hear me?
Yeah.
Okay, it's Shabtai Coin. So you have aquaporins and you have lacunae and other things changing the conductance, and actually hydraulic conductance of roots is very variable and changes with a lot of things. And I just wanted to comment that we also measured changes due to differences in relative humidity in Arabidopsis especially, and these were not influenced. There were large changes at different relative humidities. So we did 17% and 80% relative humidity. And we saw large differences in hydraulic conductance that are probably in the root system. We also, when I worked with Bill Tyree, we also saw circadian rhythms in excised roots in the dark in a pressurized system. And these would last for several days after cutting the roots. So there were differences between day and night that were circadian, that were internal rhythms. And that may be part of what you're seeing in your figure that showed the daily curve. That daily curve may be internal rhythms of the plant. Anyway, that stuff, that material of Mel Tyree's was never published because he said he didn't understand it, and we did a number of experiments like that, but it was never published. But other people have published about circadian rhythms in root hydraulic conductance. And it was in the dark. We put it in a dark room in a pressurized system. It was amazing.
Okay. Yeah, that's totally cool. I have two. It makes me think of two things. One is that it's amazing to me we don't publish a lot of stuff that we do with root hydraulics for that reason. Like your story about Mel Tyree. These pressure flow relationship curves, we built these up. I built up many of them, these sigmoidal curves of using hydrostatic pressures. We didn't publish any of it. I was a lot younger then, and maybe I'm slightly ashamed now, because when we don't publish stuff because we don't understand it, I don't know if that's the best policy. I just wanted to say that I think we should feel more comfortable publishing results that we don't understand. The journal's editors and reviewers make this very difficult on us, and it may be one reason that would push scientists to leave that system. But the circadian thing is really interesting. I'll say that when Lee was doing her PhD work, one of the things that still intrigues us maybe more than anything is we saw a lot of cyclic growth patterns in the rhizotron experiments, because she did amazing work, amazingly detailed work with quantifying growth rates, and with tons of data, hundreds and hundreds of roots. And there is a lot of rhythmical change, and it's just, we can't, I think because our measurements don't have a time resolution that's high enough to capture it, again, we then ignore it. So those are good points.
Thanks.
Anybody else? Any questions for Greg?
No, thank you.
I have a question. So you showed us the response of root hydraulics to stress, and it kind of approached, you showed a very fast decline, but then there's always some conductance, right? You never measured the zero. And I assume because, well, you probably only measured the functionality, right? Functional ranges which plants actually thrive, and they do need some conductance. But I'm curious, what do you think is different between these two ranges? The range which is very dynamic, and the range which always show us some marginal conductivity.
Yeah, I think, like, traditionally, I think I can't decide between these two explanations, basically. All right, like, one could be that it doesn't really matter out there. There's always some residual sort of conductivity of the root, because by that time the root is kind of shrunk and it's disconnected itself from the soil water. So it's kind of like fused in a different way. It's fused at the root soil water interface, and it doesn't really, the rest of it doesn't really matter. There's some residual conductivity there. Because remember, everybody needs to remember that when we're measuring this stuff, we are overcoming that with the measurement, because we're instantly pushing on it with some pressure to get the water to flow. so that could be one explanation, but the other explanation could be that the between aquaporins and just apoplastic pathway regulation, and I don't want to get into it too much because my ideas on it are so fuzzy and not well formed. But if you can imagine like water moving through a tissue, if it's moving through the spaces in between the tissue, living cells, you could imagine that the conductivity of the apoplastic space could be regulated by the cells that surround it. It could be regulated in part by the cells turgor that surround it. So when you get a collapse, a total collapse in turgor, you have no aquaporin activity left. You have something that is analogous to a minimal conductance, and it's just what's left through the apoplastic space, and it's always going to be there.
Thanks. Yeah. Any last question for Greg? Okay. So thank you very much.
Yeah, thanks, Uri.
It was
Anytime.
Really great talk.
Well, thanks, and always feel free to email me if you have a question. But thanks for the opportunity to get me thinking about this.
Okay.
Okay, thanks.
Bye, everybody. Next week we have Rob Skelton, which will also give us a great talk about hydraulics. And
Neat.
see you next week.
I'm waiting for that.